This technology primer is about blood transcriptomics. The researchers conducting blood transcriptomic studies measure leukocyte RNA abundance on a genome-wide scale. Transcription in human cells is a dynamic process in which DNA is used as a template to produce a wide variety of RNA species. The collection of all the RNA produced in a cell or population of cells is known as the transcriptome. Leukocytes that circulate in the blood can be a source of valuable information about the health status of an individual. They respond to immune stimulators, for instance pathogens, as well as endogenous processes such as inflammation. This video shows in a mouse blood neutrophils stained in green leaving the circulation to populate the surrounding inflamed tissues. Leukocytes can also in some circumstances migrate from tissues back into the blood circulation. Abundance of all transcript species present in a sample can be measured at once using microarray or RNA sequencing technologies. Microarrays consist of 10 of thousands of different probes immobilized on a solid surface. These probes capture complementary labeled oligonucleotide sequences. The intensity of the fluorescent signal produced is proportional to the number of transcripts that are present in the sample. RNA-seq relies on next-generation sequencing technology to elucidate the sequence of short nucleotide fragments derived from the RNA species present in a sample. These fragments, also called reads, are aligned against a reference genome. The abundance of a given transcript is determined based on the number of reads that align to it. The more reads align to a given gene, the more abundant is the corresponding RNA species in the sample. In clinical studies, blood transcriptomics can be employed to assess the immune status of individual subjects, in health and disease. Measuring RNA abundance in whole blood can identify changes in transcript composition associated with disease processes. For instance, changes have been identified in the blood of patients with active tuberculosis disease. On this heat map plot, the abundance of transcripts is represented by different colors. Red indicates an increase in abundance compared to healthy controls, and blue a decrease. Transcripts are organized in rows and patients in columns. The heat map shows that the more severe the disease, the more perturbations can be measured by blood transcriptome profiling. RNA abundance can also be measured in fractionated leukocyte populations, such as monocytes, T or B lymphocytes, neutrophils. And RNA abundance can also be measured in single cells. Another application of blood transcriptomics is as readout in vitro blood stimulation assays. For instance, these stimuli can be pathogen-derived molecules and other host-derived immune factors. This allows measurement of the competence of the innate immune system. Such approach was used to measure residual innate immune competence in patients with primary immunodeficiencies caused by a loss-of-function mutation of the IRAK4 and NYD88 genes. As a result of these defects, patients are especially susceptible to invasive pneumococcal disease during childhood. When blood of these patients was cultured with a broad stimulator such as PMA, similar transcriptional responses were seen in patients and controls. But when using pathogen-derived molecule, the defect in the ability of the blood leukocytes of those patients to respond became obvious. This was the case for the 500 genes induced by LPS and controls, and especially for the 237 genes induced by PAM2, which showed virtually no sign of changes in RNA abundance in patients. This concludes this technology primer on blood transcriptomics. Many other use cases of blood transcriptomics approaches can be found in the scientific literature. To learn more, see the links in the description of this video.